Ik heb toen ook stiekem wel een beetje gelachen. Dat klopt dus hoor.
According to the customs of this university, we open this session with the following words: Spiritus Sancti Gratia, illumina sensus et cora nostra. Please take your seats. Welkom hier in de aula van de Radboud University Nijmegen, especially the promovenda, her parents, welcome. Welcome also on the people who follow this session by live stream. And welcome to all the people here. My name is Paul Stuit. I was an internist affiliated to the Radboud University Medical Center. I was professor here, and then you have the honor to replace the rector, and on this moment that is Professor van Krieken. And my task is to lead this ceremony. And therefore, I give the floor to the promovenda.
With the permission of the Doctor Board and in order to obtain the degree of doctor from Radboud University Nijmegen, I would like to defend in public my dissertation entitled Multiomics Data Analysis to Understand Hemostasis and Inflammation in Healthy Individuals and in People Living with HIV, Studies from the Human Functional Genomics Project. As a human beings, we are all unique in our own way, and our individual traits and characteristics are determined by a complex interplay of both genetic and non-genetic factors. Our genetic code, which is specific to each of us, plays an important role in shaping who we are, from our physical appearance to our susceptibility to develop certain diseases. In addition to genetics, non-genetic factors such as geographical area where we live, the seasonality of our environment, our dietary pattern, as well as the exposure to the infectious and non-infectious diseases can all influence our biological traits. Understanding the complexities of our biological trait is a crucial goal in the biomedical research. One approach that has shown promise in this area is multiomics data analysis. Omics refers to a vast set of molecules with similar biochemical properties, including genomics, which studies the genome, epigenomics, which studies chemical modifications to DNA, transcriptomics, which studies gene expression, proteomics, which studies proteins, metabolomics, which studies metabolites, and microbiome, which studies microorganisms living in and on our bodies. By using holistic approach of multi-omics data analysis, we can gain insight into a novel or less known biological pathways, developing new drug, and identify biomarkers. Overall, multi-omics data analysis can pave the way for the future of precision medicine. My PhD research focuses on the use of different omics spectra, including genomics, proteomics, metabolomics, and gut microbiome, to understand the variation of both hemostasis and inflammation in healthy individuals and in people living with HIV. Hemostasis and inflammation are critical biological processes that help to maintain our body's health, and gaining a deeper understanding in both hemostasis and inflammation can pave the way for developing the precision medicine. During my PhD, I had the opportunity to analyze multiple omics data from various cohorts of Human Functional Genomics Project. In the first part of my thesis, I analyzed data from several cohorts of Dutch healthy individuals of Western European ancestry, including 500 FG, 200 FG, and 50 FG. In addition, I also extended my analysis into the inclusion of 300 healthy Tanzanians of Eastern African ancestry in the so-called 300 Tanzanian FG. The number in each cohort name represents the total number of the individuals included in each cohort. In the second part of my thesis, I analyzed and specifically into the individuals living with HIV, including 200 HIV cohort and the 2000 HIV cohort. In the first part of my thesis, I focused into cohorts of healthy individuals. During my research, I performed multi-omics data analysis to understand the variations of functional hemostasis assay measured by platelet reactivity and thrombin and plasmin generation data. First, I assessed the variation of platelet reactivity using metabolome and gut microbiome data, and the variation of thrombin generation using genomics data of Dutch healthy individuals. In addition, I also assessed the differences of thrombin and plasmin generation between Dutch healthy individuals and Tanzanian healthy individuals using genomics and metabolomics data. One example, using genetics data, I identified a genetic variant located near KLKB1 genes that was associated with prothrombotic trait. These might help to explain or provide a mechanism into the previously reported association between KLKB1 gene and a deep vein thrombosis. While there have been many multi-omics studies conducted in recent years, most of these studies focus only in population of European ancestry. One of the striking examples is that only 0.2% of the total genomics association study performed in the population of African ancestry. In this research, I compared the hemostasis and inflammatory parameters between Dutch healthy individuals and Tanzanian healthy individuals. Interestingly, I found that Tanzanian exhibited a procoagulant and pro-inflammatory state compared to Dutch healthy individuals. To understand the underlying mechanism behind this observation, I performed genetic association study with the hemostasis parameters in both Dutch and Tanzanian cohort. I found limited overlap of genetic regulation for hemostasis in both of the population. These findings underscore the importance of considering population diversity when conducting omics-based studies. In the second part of my thesis, I focused into cohort of people living with HIV. After the introduction of antiretroviral therapy, or ART, people living with HIV are almost having similar life expectancy as uninfected individuals. However, they're still experiencing a higher risk to develop certain comorbidities, including cardiometabolic diseases, malignancies, and neurocognitive disorder. The exact mechanism responsible for this phenomenon aren't fully understood. However, chronic immune activation and microbial translocation are thought to be major contributors. To better understand persistent inflammation in people living with HIV, I compared 92 plasma inflammatory proteins between people living with HIV from 200 HIV cohort and a healthy control from 500 FG cohort. I found that people living with HIV on long-term treatment still having an upregulation of plasma inflammatory proteins. Moreover, I identified subset of people living with HIV with high inflammation that were 3 and 1.5 times more likely to develop malignancies and cardiovascular diseases during 5-year follow-up. These findings indicate that a more precision approach to medicines is needed, rather than one solution fits for all paradigm to intervene with the inflammation-related comorbidities in people living with HIV. During my PhD, I was fortunate to be part of the 2000 HIV study. 2000 HIV study is the biggest multi-omics cohort of people living with HIV that were enrolled in the 4 different hospital centers in the Netherlands. This cohort comprised of a full set of multi-omics data ranging from genomics, epigenomics, transcriptomics, proteomics, metabolomics, and microbiome data. One example, by analyzing almost 1,500 plasma proteins in people living with HIV and compare it to the healthy control, I observed a dysregulation of immune and metabolic pathways in people living with HIV and highlighted the importance of gut as a source organ of these dysregulated pathways. This is just the beginning of what we can learn from the 1000 HIV cohort, and I'm excited to see what the future holds. To summarize, overall, in this thesis, I demonstrate the significance of multi-omic analysis in comprehending pathways in health and disease, specifically into the regulation of hemostasis and persistent inflammation of people living with HIV. For the future, I believe that multi-omics data analysis that take place in the 1000 HIV cohort will shed a further light into the complexities of HIV infections, including unique phenotypes, comorbidities, and latent viral reservoirs. These could pave the way for precision medicine and even a cure for HIV. Furthermore, we identified differences of important biological processes between individuals of European and East African. These highlight the importance of performing multi-omic studies in underrepresented population. As coming from Indonesia, I also support fellow researchers from the Global South to take part into making multi-omic studies more inclusive. Having presented the summary of my doctoral thesis, I return the floor to the rector.
Thank you, Ms. Nadira. And we start now the opposition. I will explain you some details about this ceremony. Next to me, you see the first promoter, Professor van der Ven, and on the screen, in Indonesia, you see the second promoter, Professor Gazem. Next to the first promoter, the 2 co-promoters, Dr. Matsaraki and Dr. De Mast. They supervised the work of the candidate, so they are not supposed to ask questions today. Next to the co-promoters, you see the opposition. I will introduce them at the moment they get the floor. And behind me, you see 3 professors for encouraging the candidate. That's Professor Dolmans, Professor Joosten, and Professor Netea. The first opponent is Professor van der Vlag. He is affiliated to our university as professor in immunology of renal diseases. He was also chairman of the reading committee. The floor is yours, Professor van der Vlag.
Thank you. Dear candidate, first of all, I want to congratulate you with this excellent thesis. It's one of the best theses I have ever evaluated, so you can be proud. I also want to congratulate your supervisors with this fantastic work, all done within 4 years. Very good. Normally, when I evaluate a thesis, I always make already notes for possible questions, but in your case, I almost had no questions, so I was a bit desperate. So I will start with some general questions. So you introduced very nicely your thesis, and you mentioned the multi-omics approaches, different ones, all the multi-layers. Mm-hmm. But I think an important layer is missing. Can you guess maybe? Yes. Which layer?
Dear opponent, thank you for a very nice compliment. I think the missing layer will be the functional validation.
Yes, we will come back to that layer later. But now I was hinting on glycoprofiling. So, so the complexity of our body is mainly dictated by glycosylation. Mm-hmm. And so that was maybe for a follow-up project, and adding another layer. Then, yeah, I want to discuss briefly with you chapter 4 and 5, and you compared healthy cohorts from Europe and Africa. So I was amazed that there were so much differences. And even within the African cohort, you had 2 subpopulations, and you suggested that was caused by the feeding pattern, depending on the season. And so at a certain moment, I wondered, what is now the definition of a normal, healthy cohort?
For the normal healthy cohort, of course, what we define here is a cohort without any prior comorbidities or current comorbidities. And specifically for the cohorts in Africa, for example, we really make sure that there was no comorbidities also with malaria infections, also with HIV infections, so we also tested that. And in regards to the cardiometabolic diseases, for example, we also measured a blood glucose levels, random blood glucose levels, also the blood pressures. So overall, we just make sure that these healthy individuals is as healthy as possible as we could.
Okay, so that is a kind of definition then.
Yes.
So you have some criteria. Mm-hmm. But I wondered now with this new knowledge, mm-hmm. how we should select a normal healthy cohort. For example, when you look in this tiny country, mm-hmm. the Netherlands, you already see a gradient in different HLA mm-hmm. from north to south. So should we not come up with better criteria for a normal, healthy cohort than blood pressure?
Yes. So for molecular profiling, for example, when we defined a subset of cohorts of population as a healthy, we still need some kind of guidance. So we have to pre-select it beforehand before we can actually perform molecular profiling of them to understand whether they're truly healthy or they're actually experiencing a little bit of inflammations compared to the other populations. I would say it's a challenging task, of course, and we don't know whether the definition of healthy for one people would be similar to the other people, because my blood pressure can have 120. For the others, it won't be necessarily to be the case for them being healthy. So it is a very challenging task, I find. But of course, we can make use of the technology that we have now to better define healthy.
Thank you.
Yes.
So it is challenging.
Yes.
I agree. So I can move on to chapter 6. I see no signs. So in chapter 6, you describe that plasma serotonin and indole metabolites are associated with gut microbial composition. You describe a lot of associations, and that is what you already mentioned. So you made a fantastic drawing on page 168, in which you summarize a lot of mechanisms. And I wondered, first of all, the context of your cohort. So they are treated with antiviral medicine, mm-hmm. and they have an underlying disease. So I was wondering, that was my first question, what is now causing these relations? Is it the medicine or is it the underlying disease?
Yes. In the chapter 6, we try to explain an association between gut microbiome and tryptophan metabolisms, also in regards to their functional consequences, possible functional consequences. And you are correct that we're only looking at the population of people living with HIV. There might be obscured also the true association with the multiple factors that might influence them. For example, in the microbiome, we can think about diet, for example, or even the lifestyle. People with HIV might also be different with the healthy populations. And as you also mentioned, for example, the medications or their comorbidity. So they're very complex. And what we show in the chapter 6 is merely an association of a cross-sectional study. So we try to find a pattern. We try to see the overview, how in the population with such a dysbiosis in their gut, in their microbiome, could somehow affect their metabolism and in return to their phenotype as well. Of course, for the future study, we really need to have like a clear-cut definition. So we want to dissect whether this is something that influenced by the medication itself or by the virus, or even comparing with the
Okay,
health.
thank you.
Yeah.
So when you look at those, the scheme you draw on page 168 and
Yes.
your suggestion about the microbiome, what would be a good experiment to test whether the microbiome is essential or not?
I will say what comes to my mind is the mice model so far. So when you have like a germ-free mouse and then you try to give their microbiota, I mean like the microbiota from the other mouse to this mouse. So it's like gaining function or losing function experiments to really test whether the microbiome or selected microbiome that might produce a certain tryptophan metabolites could really influence their phenotype. If you want to test like it is in the context of psychiatric symptoms, then you measure psychiatric
Very
symptoms.
good.
Yes.
Very good
Something like
experiment.
that.
And what you also could consider in patients, an intervention with fecal transplantation.
Yeah. 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 Exactly.
Okay, thank you very much for your very good answers. I give the floor back to the rector.
Thank you, Professor van der Vlag. We now go to Professor Lee. Professor Lee is from Germany, Hanover, the Helmholtz Center for Infectious Research. She is professor in computational biology. She was also a member of the READY committee. Thank you for coming all the way from Hanover. Professor Lee.
Thank you very much. Dear candidate, congratulations on completing such a beautiful thesis. I enjoyed reading very much. Also, congratulations to the supervisor team. So, now look at your thesis, chapter 1 on page 14. You stated that it is important to perform thorough pre-processing quality control of raw data, especially try to control the bias in the data sets. As a computational biologist, I cannot agree with you more. Then it comes to the questions. In chapter 7, you show that in table 1 and table 2, age, gender, smoking are associated with your phenotypes. Then my question is, how did you address this potential bias from those factors? And what steps did you take to control such effects?
Dear opponent, thank you very much for the compliment. I think it is really, really important to perform a thorough pre-processing steps before going into the downstream data analysis. And for the chapter 7, for example, in the first place, we always look at the possible confounders, especially when we try to compare like different cohorts. Therefore, when we look at the demographic, for example, age, sex, BMI, or even smoking status, we saw differences between the populations, and we try to address that actually using a linear model. So we introduce them as a confoundings, the possible confoundings, and when performing differential expression analysis. And in addition, we also tested whether having a matching samples, so matching samples with the age, sex, and smoking status, will actually make any differences in comparison to the adding them into the linear model, and we saw a consistent pattern.
Mm-hmm. So maybe another step after you identify those significant proteins or metabolites, did you ever check whether those molecules that are associated with age or gender? Because this could help you to avoid this potential bias. Do you think this is a necessary step or not?
Yes, for example, we can try to stratify it, for example, based on the top hits, top hits of the molecule that might be very important in our research. I think it's very nice suggestions to actually double check that after with the stratifications, because now we already corrected them in the beginning, and we want to see where there is a remaining effect left of the demographic in the comparisons. Yes.
Okay, a very small technical question. So you use a smaller cohort as a discovery in chapter 7,
Yes.
but the bigger cohorts as a validation. Do you have a specific reason for that? Because this is contradictory to what is usually done in the field.
Yes, usually people will use like a bigger cohort for discovery and a small cohort for validations. However, at that time, 1000 HF is still at the enrollment phase. We only had like the first 600 of the participants, and the dataset was also not complete enough as compared to the 200 HF cohort. However, we did try to actually switch the discovery and validations approach. So we tried to put the emphasis on the discovery cohort for 2000 HFE and then make a validation 200 HFE, and the results is comparable. Therefore, we go with the first approach. Yes.
Very good, thank you. Yeah. So if time allows, I will continue with the next question in chapter 4. So here you demonstrate that the season effect is very important. However, in your model, you didn't include it. But you only include age and gender. What's the reason for that?
So the reason is the first we see as a major confounding is always age and sex, especially for the nature of the cohort when we compare 500 FG and the Tanzanian cohort. The influence of the season and the influence of the other environmental factors, we think it is more into something that we actually want to discover. So we want to see the influence of difference between environmental factors in both populations, how they can actually contribute to the differences in the phenotype itself. So therefore, we don't correct for that in the beginning when we compare the overall comparison of hemostasis parameters.
But do you think this unwanted variation will reduce the power for genetic analysis in the next step? Because later on you show there is a limited overlap in the genetic regulation of TG and PG. So do you think this seasonal effect could potentially, reduce your power to see the genetic correlation between them?
Yeah, that might be also reducing the power. Yes, I agree. But the thing is, I'm also thinking on another way that the fact that they have such a different environment that's shaping their genetic regulation to a certain biological processes might also be of a focus there. But yes, we can also perform an additional analysis to see whether the effect stays after the correction of seasons. Yes.
Yeah, I see. Thank you. And I think in your thesis, you are not only demonstrating the power of multi-omics in immunological research, you also nicely show how multi-cohorts can be integrated together to gain the novel insights. And in your introduction chapter, you classified the multi-omics strategy into 5 categories. I was wondering, which category of the methods did you use in your thesis? And which method should we, in practice, select for our research questions?
In my thesis, I mainly use a late integration approach. So basically analyze the omics per layer and then try to find like a similar pattern between the omics layers. But as a future, while the field's still developing, I believe that perhaps the best approach is to start with the genome. So we start with the blueprint of our life and then start to layer it by sequential of layer. I mean, like from the genome, and then you can go with the epigenetic and transcriptomics. But of course, there's multiple ways to integrate the multi-omics studies, and it depends on what questions that you want to answer. For example, if you want to stratify the people based on their molecular profile, maybe you will go with the factor or Bayesian factor analysis. But if you want to know the pathway or the mechanisms behind such observations, then you'll go integrating it layer by layer.
Very good, thank you very much. last question. So you in chapter 6, 7, and 8, you nicely compared the multi-omics data sets between HIV and control. Can you reflect on the overall impact of your research on the fields of HIV studies? And what direction do you see in the fields moving in the future?
Yes.
Thank you.
So what I see when comparing people living with HIV on long-term treatment with healthy control is that without that, they'll be very much similar to the healthy control. Well, in fact, it's not. So we see a huge difference, such a systematic difference of protein expression levels in the people living with HIV on long-term treatment with the healthy controls. So we think, so for me, I think that it might be wise to also look at those with the persistent inflammation or those with having a high level of inflammations compared to the others, whether they really need an additional treatments to alleviate the inflammations or perhaps not, basically to increase the quality of life of people living with HIV.
Right. So you already have 2,000 HIV
Yes.
samples. Do you think in the next 5 years we should go for 10,000, or what's the next steps?
The next step, of course, in the genetics, for example, the more the samples, the better, because you get bigger resolutions and you have the power. Because the question of omics is always like, do we have enough power to capture an association, or we just have so little power that we cannot see it? But sometimes having a little more of a participants also computationally expensive and also hard to work with, but of course it depends on the questions, and it also depends on the technologies that will be there in the future. So yeah.
Very good. Thank you very much. Now I return back to the director.
Thank you, Professor Lee, and thank you for coming all the way from Hanover, and thank you for your membership of the reading committee. The next opponent is Dr. Scholz. Dr. Scholz is an internist affiliated to the Radboud University Medical Center, the Department of Hematology. The floor is yours, Dr. Scholz. He was also a member of the reading committee.
Thank you very much for the introduction. Dear candidate, dear Nadira, first I would like to congratulate you and also your supervisor team, colleagues, friends, family, with successful completion of your manuscript. It was really a joyful moment for me to read your whole manuscript. As a clinician, as an internist hematologist, I always try to find the clinical implication of research. And by saying that, I would like to discuss with you the results of Chapter 2. Mm-hmm. In Chapter 2, you have performed an untargeted plasma metabolome study and got a microbiome profiling with the aim to provide novel insights and platelet reactivity in healthy controls in Chapter 2. And especially on page 40 in the second paragraph, you mentioned that in the analysis on the correlation of individual metabolites of the untargeted profiling with platelet reactivity, the most prevalent positive associations were found for metabolites belonging to the class of the lipids, and negative associations were found for the organic acids belonging to the category of others. You can also see that in the page adjacent to it in 41, figure 1. Can you explain these results? And is it possible to translate these results for clinical practice? Can we use these results? Mm-hmm. And do you think that these results can a little bit be modified by the way you actually measure platelet reactivity?
Yes. Dear opponent, thank you for the compliment. I'm thinking for the Chapter 2, we try to understand the regulation of platelet reactivity using metabolome and gut microbiome data to see whether we can find a less known pathways that regulate the platelet reactivity. And yes, we found that not only lipid, but also amino acid metabolism might play a role in the platelet reactivity. For example, in regards to lipid, we found arachidonic acid metabolism as a top pathways that also highlights the results of our analysis with the use of aspirin as a widespread of antiplatelet therapy. And in regards for amino acids, for example, it's very little known about the amino acid metabolism and platelet reactivity. Often when I try to understand the process better, it's always obscured by how little we see the association of individual metabolites with platelet reactivity. So sometimes you have people comparing amino acid with little or too much or supraphysiological concentrations that also making the interpretation a little bit difficult. However, I think in the future the application of this result is a stepping stone. So if it's the best line, And this is also a correlation. And the dynamic of the metabolism itself, like longitudinally, the stability of it, and whether it is really reflecting a causative association or not, still needed to be known before we can step it further into direction of therapy, for example. But for now, I think I can highlight the importance also of the gut microbiome here, that surprisingly also play a role in the reactivity of the platelets. And I think, for example, such as simple implementations like a dietary restriction or perhaps taking a probiotics might have results, but of course needs to be validated in the future studies. Yes.
Thank you very much. And can you also comment on the methods of platelet reactivity, why you choose to measure the p-selectin expression and the binding of fibrinogen to its glycoprotein?
Oh, yes. So we try to use the strongest stimuli for measuring the platelet reactivity, and that's the CRP. So we try to capture as much as the variation as we can in this cohort, especially in the young healthy individuals. And of course, using a different platelet method, maybe we can find like a different pattern. So that's also something to think about. But at least we see a consistency using flow cytometry data between 4 different measurements that we took. So there is a strong internal consistency between them that reflect a good measurement overall, and perhaps we can trust the results. Yes.
Okay, thank you very much. In my way to find the translation for clinical practice, I would like to take you to page 43, 2 pages further on. In the middle, you state that you postulate that the higher levels of androstenedione, androstenedione, and testosterone in males in your cohort contributed to the higher fibrinogen binding compared to females. Can you use this information to create a different approach for cardiovascular research management for males versus females?
I would say to give like more emphasis and monitoring therapy for the treatments. For example, we can also take a look into their hormonal levels, whether it is actually contributed to platelet reactivity, yes or not, or whether the levels of the metabolites itself could be reversed or could be increased, for example, in regards to their platelet reactivity profile, depending on the disease that we treat. But I feel like we really need to look into this further as again, we found such a correlation, so I was a bit hesitant to move forward with it. Yes, it's just an observation, one-time thing, and of course, it needs to be validated. Yes. Okay.
Can you propose a study proposal? Can you set up a study proposal in which you can determine which kind of biomarkers will be best for evaluation cardiovascular risk management in males and females?
The studies, I would say to include like healthy individuals, for example, or diseased individuals, and then we can stratify them based on sexes, and we can also measure their hormonal levels and also the platelet reactivity or other measures of cardiovascular diseases. In the omics, perhaps I would measure the as much as omics as possible, but if I can choose, I think the closest to the phenotype is the proteomics, so perhaps proteins can also shed the light into these differences and then can pinpoint the biomarkers since it's easily translated to the clinics.
Okay, thank you very much. If I can ask another question. On page 46, in the middle, you state that the serum gamma-GT has been correlated with an increased mean platelet volume, and that the gamma-GT is present in the platelet membrane and can serve as a predictor for cardiac events in patients with an acute coronary syndrome. And can you speculate what comes first? In Dutch, we have a very known word for that. What is it kip and what is it ei? Can you found a direct causal relationship between the serum gamma-GT and cardiac events?
Yes, to understand whether this is a causative correlation, yes or not. At least in the analytical step, I will do, for example, Mendelian randomization analysis. So in this cohort, I don't have the phenotype, for example, the cardiovascular disease, to actually see whether this is a causal or this is actually consequences for this observation. So I would say like doing a genetics, for example, and to see whether a certain genetic loci is associated with the both phenotypes will really tell us whether this is a causative or not, and then we can, move forward with it.
Yeah,
Yes.
okay, thank you. And if I can ask the final question.
Absolutely.
Yeah, it's on page 50, the last sentences of your discussion. You say that finally our study population consisted of predominantly young individuals, and this may limit generalizability of your findings to older individuals. Can you explain in what direction that young individuals could have modified the results, especially the environmental factors?
In this analysis, we tried to also correct for age. So with the correction itself, the idea is to disregard the effect of age itself in the analysis, so it should be translatable to people of all age. However, of course, an overrepresentation of certain age category will also obscure the associations, since we cannot capture their variability in the analysis. So I would guess that having distributions of age from every category will help us to understand this more. If I can speculate, I would say that there will be, of course, difference because of the aging process, as we know that aging not only take a toll into the reactivity of the platelets, but also into the immunological state that's closely related to the thrombosis. So yeah, I would say that there might be difference, and it needs to be further studied. Yes.
Thank you very much for the clear answers, and I will give the word back to you.
Thank you, Dr. Scholz, and thank you for your membership of the reading committee. The next opponent is Dr. Blok. He is retired internist, he was affiliated to the Onze Lieve Vrouwen Gasthuis in Amsterdam. The floor is yours, Dr. Blok.
Thank you, Professor Stuart. Dear candidate, first of all, I would like to congratulate you on your thesis, and I think it's very well written. I'd like to also extend my congratulations to your family and friends and to your promoters and co-promoters. It's a very interesting thesis. I have enjoyed reading it. Very clarifying figures. So this kind of work also brings some questions, of course. As a clinician, I would first like to go to chapter 2, metabolome, microbiome, and platelet reactivity. As a clinician, I was very curious about the possible role of the metabolome and the microbiome on platelet reactivity, since it's such an important feature in atherosclerosis and indeed in acute coronary events. So my question concerns the finding of the correlation of the arachidonic acid pathway products, such as prostaglandins and leukotrienes, and platelet reactivity as you depicted in figure 1 on page 41. I was happy to notice that you performed dietary questionnaires in your study group, and you did not find any correlation between the consumption of fruit, vegetables, alcohol, and caffeine on platelet reactivity, as shown in supplementary figure S4 on page 56. Now, about 30 years ago, one of your co-promoters, who is present in this room, Professor Netea, was involved in research on the influence of fish oils on all kinds of biological parameters. Therefore, I was a bit surprised to notice that you didn't ask the participants whether or not they consumed fish. So given the fact that fish consumption, especially fatty fish, and thus omega-3 fatty acids, changes the arachidonic acid pathways in a way that could potentially influence platelet reactivity, would it have been informative to ask for the ingestion of fish?
Yes.
What do you think?
Dear opponent, thank you for your compliment. In regards to the questions, yes, we did ask for a fish consumption. In the way of the analysis, I really need to do a pre-processing of the data, and I realized that there have been a lot of missing data, for example, in the fish consumptions. Therefore, I'm not including them as a supplementary, as we didn't totally trust the results at that time. However, of course, the variation of the metabolome is also hugely influenced by the diet, but also some metabolites can be produced endogenously. For example, the arachidonic acid metabolism for the platelet membrane or for the other phospholipid membranes in the body. I believe that such studies, for example, to also taking into account the specific dietary requirements like omega-3 or like other dietary how to say, like regimens would be very important to understand whether this specific metabolite, especially the arachidonic acid metabolism, really change the way we ingest this omega-3 or other dietary regimens. So yes, might be very important. However, what I found, like in most of the studies, I think such a confounding effect is huge that it's sometimes hard to see like a direct association between diet and the metabolome, for example, that translate also for the reactivity. So it's always vary between studies and hard to, yeah.
Thank you. Dietary studies are sometimes really difficult, I
Yes.
know.
Mm-hmm.
Now, my next question also relates to diet, I'm afraid.
Mm-hmm.
In chapter 4, you describe elegantly how thrombin and plasmin generation potential differ between the Western European cohort and the Tanzanian cohort. The Tanzanian cohort consists of 323 participants, 70 of which live in rural settings on the slope of Mount Kilimanjaro. Now, on page 103, you state that you did not find any difference in thrombin and plasmin generation parameters between the urban dwelling and the rural dwelling Tanzanians. However, you did notice seasonal differences in thrombin and plasmin generation. And you hypothesize that these differences are related to differences in diet. Previous work by the same group, and you refer to the paper by Temba et al. In 2021, found differences in inflammatory status driven by dietary differences between rural and urban living Tanzanians. So my question is, given the fact that the difference in biological parameters between rural and urban dwelling Tanzanians is mainly driven by differences in diet, and that seasonal differences are also driven by diet, how do you explain that you did not find a difference in the thrombin and plasmin generation between these two groups?
Yes. For the questions, we actually, for the previous publications by Temba, we actually found such a huge differences in the ex vivo cytokine production capacity. So it's the functional immunological response. For the chapter 4 and 5, while the coagulation and inflammation is closely related to each other, we found no differences between the urban and rural population in regards to the coagulation, also inflammatory process, but it is baseline inflammations. So we think that perhaps what differs between them might that also confound by the seasons and the diet. It's actually the production capacity and not the baseline level. So that's why we couldn't find differences between urban and rural and Tanzanian cohort for the thrombin and plasmin generation.
Or could it be that these rural dwelling Tanzanians secretly do take some processed food? What
Yes. It might be late, yeah. It might be also, but for example, during the certifications,
Yes.
while we try to find like two separate endotype of the metabolome, we actually see a similar number of rural and urban participants between two categories. So we think that perhaps they can snake, but of course, perhaps the frequency is similar to those in urban. Therefore, yes, underlined also our conclusion that there is no difference in the urban-rural population. Yes.
Thank you for your answer. Now, in chapter 5, you describe differences in the inflammatory proteome of a Tanzanian and a Dutch cohort. And you relate these differences to environmental and dietary factors. In your introduction, on page 134, you mention the fact that urban living Tanzanians display a pro-inflammatory gene signature. And you suggest that the historically high burden of infectious diseases in this group may have resulted in the selection of genotypes favoring a robust immune response. So vice versa, I was thinking, in people of Western European ancestry, and maybe a relatively anti-inflammatory genotype would have been selected. Now, I was wondering, given the fact that the burden of infectious diseases in Western Europe only started to decline since 1850, so that is about 170 years ago, or let's say 8 to 10 generations, would such a short period of time be long enough to account for such a profound selection of genotypes? What do you think?
Yes, I don't think that such a time would actually, it will contribute to certain changes, but not to the extent that we expected, of course. In regards to that questions, I think that it's not only the genetic regulations that might differ, not only the positive selections in regards to also the inflammatory burdens or infectious disease burden between the two populations, but I think it would also be very possible that the public health approaches would also play the role, and also there are seasonal variations that might be differences, or the diet that we see like more monotonous diet here in the Europe, while we don't see that in the Africa, while we have like such a huge variety of foods that we can take. So perhaps there's other factors that might influence the differences, but not only the genetics, of course.
Yeah, 7 to 10 generations may
7
be
to 10 generations, yes.
too little, I think,
Yeah.
for these kind of mutations. Okay.
Yeah.
I have a question on your chapter. It's more a philosophical question. It's on chapters 7 and 8. You work in people living with HIV.
Yes.
Now, in the introduction to these chapters, you refer to the fact that people living with HIV, despite the fact that ART has dramatically improved their life expectancy, that they still have some excess incidence in cardiovascular disease and also non-age-related cancer and psychiatric illness. And this has all been related to the so-called chronic immune activation that persists in these patients despite viral suppression. I think understanding the origin of this immune activation and hopefully finding a solution to overcome it remains a holy grail in HIV medicine. One hypothesis that you refer to and brought forward by investigators such as Jewett and Deeks, Brenchley and Hunt holds that early in HIV infection, lymphoid tissue lining the intestinal tracts is destroyed, thereby destroying the intestinal barrier, and this is the so-called leaky gut hypothesis. On the other hand, you find, and others with you, that there is also a matter of intestinal dysbiosis in people living with HIV, perhaps also leading to immune activation. So maybe you can elaborate a bit on these two phenomena, the leaky gut syndrome on the one hand and the intestinal dysbiosis on the other. How do they relate to each other? And what do you think is the relative contribution of each other to the phenomena of the observed immune activation? What do you think?
For the persistent inflammation in people living with HIV, people thought that there might be a complex process and multiple contributors to this phenomenon. First of all, it can be the leaky gut, but it can also the burden of the viral surfers that hidden somewhere in their body, also the art toxicities. But in regards to the gut, leaky gut hypothesis, people showed that during the early HIV infections, we actually see the dysregulations of the immune system in the gut, and gut is as the primary also organ that were infected by HIV. The hypothesis is that the changes in the immune system is also in the epithelial layers of these intestinal cells can make their permeability changes, and somehow it can also associated with the changes in the microbiome flora in the lumen. They'll go hand in hand. For example, the microbiota that will result in the dysbiosis will also produce certain molecules that might also influence how the immune cells behave, or even the epithelial cells behave, and even translate or come to the circulations and initiate the persistent inflammation. But there have been also studied for the chicken and egg that, for example, the leaky gut hypothesis that we thought is always that people come, they have the infection, and then they have a leaky gut and persistent inflammation. But recent studies also show that after controlling for huge confounders in the microbiome study in HIV, which is man sex with man, sexual behaviors, their lifestyle, for example, in longitudinal studies, they found that this biases changes were also present before the HIV infection itself. So it might also be that the microbiome somehow also affect the susceptibility of people having the intestinal dysbiosis or persistent inflammation or even into the disease. So it is complicated, I think. It's quite complicated. And yeah, I believe that longitudinal studies, for example, really need here to really dissect the timeline, like whether it is precede or is it actually more into the consequences that we all think right now. Yes.
Thank you very much.
Thank you, Dr. Blok, and thank you for coming all the way from Amsterdam. Next opponent is Professor van Krevel. Professor van Krevel is affiliated to our university as Professor in Global Health and Infectious Diseases. The floor is yours, Professor van Krevel.
Thank you very much, Mr. Rector. Dear candidates, also my compliments. It's beautiful science, beautiful empiric data, very nicely illustrated, very well written. I'm sure your supervisory team have learned a lot also. I'd like to continue in chapter 7 and 8. And I'd like to come back to, and it was also maybe alluded to in your answer, on the selection of your control group, because they're twice as old, they're different gender, their BMI is different, their smoking is different, recreational drug use, cardiovascular disease, endocrine disorders, medication, possibly ethnicity, STIs. How can you computationally correct for that?
Yes. Dear opponent, thank you for the compliment. In regards to the questions, I think that correcting for such a complicated covariates and confoundings would be too much in the analytical part. So if we want to really know the influence of each factors, that we need a really clear-cut case and control studies in which we control everything. For example, we match them based on the lifestyle, based on their diet, based on their medications, etc. But at the other hand, the selection of people living with HIV that we have is actually representative for the whole people living with HIV.
Sorry to interrupt you. Of course, they're representative for the HIV,
yes.
But your healthy controls do not match these people with HIV in any way.
Yeah. For that, we actually try to also not only to perform the correction, but at least for the demographic, because then we can correct for the demographics.
But can you correct? Because some of these relations will not be linear, and if you have hardly any overlap, let's say, if your age distribution is so different, can you actually correct for that?
So we actually try to validate the correction using the matching strategy, of course, with the cost of power, and we still see the similar analysis. So the differential expressed genes still there, and we think that with the underpower we can still find the same, perhaps the selection, the corrections actually.
You've done a magnificent job, but wouldn't it be a lot easier, let's say, to take people who go for an HIV test in an STI clinic, and who in many other terms reflect the same population, except for the fact that they don't have HIV, let's say. People with sort of same lifestyle, same age, gender, etc.
Yes, I agree. Such a study will be very beautiful, of course.
You find remarkable difference, and I started my outpatient clinic this morning, and it was interesting because you have this healthy person sitting across from you, and you speak about their mortgage or their next vacation or whatever, and you try to improve their blood pressure a little bit, and you can't imagine that their inflammatory profile is so different maybe from someone without HIV. And so my first question is, you alluded to possible factors that could account for this difference. And actually, they all sort of seem, and also within the HIV population, they seem to be coming back. So it's not, for instance, it's not the CD4 nadir, it's not the viral reservoir, it's not. What is it? Can you speculate on the fact that is actually driving these differences, both between HIV, non-HIV, and within HIV?
Yeah, we try to also look into that. We actually made like an extensive analysis in order to find why. So it's from we're starting from the demographic, smoking stages. We also looked at the comorbidities and everything. It's all round up into the history of blood pressures, having a high blood pressures before. We're not sure whether this is a sole reason that there became a pro-inflammatory.
Allow me to interrupt you again, because then I thought if it's hypertension, it would have been obvious to look for the other functional genomics cohorts that we have, where there's a lot of hypertension. Is this really hypertension?
We don't think that it's really hypertension. It might also be that this is not the cause, but this is such consequences of the changes in the moleculars in such an early stage that they became apparent after 5 or 10 years, and they develop such a comorbidities.
Okay.
For the question why, of course, we have to look at it retrospectively, and we have to measure the whole lifestyles and also environmental factors that might influence the medication, for example. Yeah.
Another interesting thing with these different protein profiles, you're trying to look for the origin of the proteins, huh? And then you come up and you use a tool, and it's called tissue. What was it?
Tissue atlas and
tissue descriptive. Well, you know the full term. What is it again?
Tissue functional enrichment analysis.
Exactly.
Yes.
Tissue functional enrichment analysis.
Yes.
Are you allowed to?
So
Comparing these populations.
yes, so that's one of the limitations, that we try to understand the origin of the proteins based on the healthy control. So the healthy control state, where are all of these proteins coming from? Is it like specifically enriched in certain organs? So we kind of make a prediction. Of course, this is not really representing something that we can directly measure, but at least this is giving us like a proxy of what might it be.
But if you already look at the architecture and your correlation matrix of things in blood, just imagine in lymphoid organs. I mean, early HIV work, for instance, people looked at tonsils, or we refer to the gut. If you look at the gut architecture, it's different. So I don't think you can predict based on healthy tissues. You can predict the origin of proteins in this population.
Yeah.
Maybe allow me to move. Let's say maybe you've made a long journey, but you come from Indonesia and you will return to Indonesia. And I was just wondering about all this work in the context of Indonesia. So the Human Functional Genomics has moved from health to disease. And so where do you think, what is the next thing we should move? We are understanding healthy, homeostasis, and now we move to disease. And what's next? What's the big next thing?
I will say that the applications of the study itself. So first of all, when you're saying about Indonesia, then I will think that, is there any functional genomic studies in Indonesia? Is it applicable? Is it translatable, what we can learn here to the Indonesian, for example? So we need to first develop such a studies in Indonesia to confirm whether we can translate the findings, yes or not. But of course, all of this analysis and hypothesis, it really needs an action to be translatable to the clinic. So we have to think of certain biomarkers or gene target.
Maybe in short, you want to move to interventions.
Exactly.
You want to improving health.
Yeah.
And I was just thinking also about the experiments on the slopes of the Kilimanjaro, and I was thinking, take it back to Indonesia. Maybe you have one group and you feed them healthy vegetables from West Java. Karedok, and you have another group and you give them nasi padang, or you bring them to the KFC, and you look at something chronic like cardiovascular disease or diabetes, very inflammatory, and you are going to hopefully solve those issues in Indonesia. Thank you.
Thank you, Professor van Krevel, for your contribution here. The defense is hereby concluded. I give the floor once again to the candidate.
Having defended my dissertation to the best of my ability, I would like to thank the rector and my supervisors and co-supervisors, as well as all those who have honored this ceremony with their presence.
The doctoral examination board retreats for its deliberations. On behalf of the doctorate board of Radboud University Nijmegen, we have decided to award you the degree of doctor. I invite Professor van der Ven to discharge the task assigned to him.
With the power entrusted by law to the doctoral board, I hereby confer upon you, Nadira Vadaq, born in Bandung, the title of Doctor from Radboud University Nijmegen, to which are attached all the associated legal and customary rights and duties with respect to academia and society. As proof thereof, I present you with this doctoral diploma signed by the rector and the doctoral thesis supervisor and co-supervisors.
Please take your seat. The laudatio will be spoken by the first promoter, Professor van der Ven.
Dr. Nadira. Oh, Dr. Oh, sorry.
Nee, dat is goed.
Is it all right?
Prima, zo.
Dr. Nadira again. Oh, Dr. Nadira, or Dr. Vadaq, we were not sure about how to address you, but we call you Dr. Nadira. It's a great honor and a very big pleasure to address you now on this new title, which you fully deserve. You are, for me, and also for the co-supervisors, a very exceptional, talented person, which has made a remarkable progress over the last 4 years. In my life, it's extremely rare to see a person developing such fast and so broadly in such a short period of time. I really admire you. So we came in contact with you by Professor Hussain and Professor Sultana, whom we collaborate already for many years. And they introduced us to us. And Professor Hussain, I'm sorry for the Dutch government, which makes all the visa regulations so difficult these days that you cannot come here anymore. And your parents, they had to do so many travel extra things to get the visa to get here. It's really embarrassing. But anyway, we are very grateful for Samarang to introduce you to such a bright lady. You sent us your curriculum vitae, and we learned that you did. You graduated from Gajah Mada University in Jogja 2014 as an outstanding student. And you received many medals, including the Student Creativity Program and also Medical Research Proposal Competition. But apart from that, I read yesterday evening, you are also a very good athlete. You can run very fast for 1500 meters. You are even the best there in Jogja. So we decided to go for an LPDP, which you were awarded. We are very grateful to the Indonesian government for giving us this opportunity and the funding so you could come to the Netherlands. And in the 4 years that followed, you wrote the 9 chapters in this thesis. And you started off with a coagulation in the Human Functional Genomics Project But then, as you got access to all this data, we felt you had a very great interest, and you expressed it also, not only in the biology part of it, but also in the bioinformatic part of that. And at that time, we started the 2000 HIV program, for which some people are sitting here in the audience. And slowly by slowly, over time, you did not only work on your own thesis, but you started also to work on the thesis of many others. Because everybody, you are a very easy, accessible person, very pleasant to work with, eager to do all this analysis. It's never too much. You always say, no, come. I will look at the data, preferably yesterday than tomorrow. And therefore, many people start finding you. Of course, together with Vicky, you made the team of the 2000 HIV bioinformatics. So before you defended your thesis, you were already a postdoc. And that is exceptional. I have never seen that before in my life. So this is the first time to experience that somebody works already as a postdoc before defending her own thesis. And why do I say this? Because this is impressive work. It's a lot of chapters. You can see that. And you mentioned in your summary that there are 14, you're participating in, you're co-authoring 40 papers. But I am sure that there will many more follow the coming time. So you can only do that because you work extremely hard. You never complain. Some of us asked for some data, then I learned you worked till 3 o'clock at night to finish the data analysis. But still the next day, very eager to get new data to analyze. So you are extremely pleasant to work this, and you do what I find also a very, very important quality. You do no concessions on what is right and what is wrong. When we sit together with all the PhDs and somebody says something, or it's me saying something and the thing say it's not correct, you say, well, maybe you can look at it like this. I think it's like that, and you can be very persistent on what you think is right and what is not right. So I really admire that. That's very good. Apart from all this work and helping all the other people, you also find time to sit in a car, to drive 3,000 kilometers to the north to look at snow. In 2 weeks' time, you go learn yourself skiing in Austria, which I think is very difficult for in a week time. And besides that, when I sit at Emmy's, then suddenly it appears that Nadira often works at Emmy in the restaurant to help her in the kitchen. So, altogether, I think you are an exceptionally talented person, a fantastic researcher, to the far the best I've ever been working with, very energetic, very intelligent, and it has been an extremely great pleasure to work with you. I'm sure you will have a great future in front of you. I hope you find a place somewhere to do a medical specialization. Of course, we hope for internal medicine, maybe infectious disease even. And my congratulations are not only for you, but also, of course, for your parents. You have an extremely gifted daughter. And when I say it's a real pleasure to work with you, I really mean it. And I want to extend also the congratulations to the people which you might look via internet in Indonesia, and to your friends and to everybody here in the audience. We are, again, very grateful for the manuscript committee for doing the work. It's always a lot of work to prepare all this, to prepare the reading and the questions, etc. And I want to thank also Professor Hussain from Samara for his continuous support. It's also a great pleasure to work with you, and I want to also thank everybody for being here today on this great occasion. Thank you very much.
I give the floor now for a few words from the second promoter, Professor Ghazem. Please unmute.
Yeah.
Yes. Okay.
Yeah. Okay. Yeah. I remember that someday Nadira come to me, and she directly asked a discussion about the possibility to take PhD in abroad, and I mean in Netherlands. I at the time was very young, and I know she was a bright medical student from Radboud University, and this is like exception. Usually, people come to. We should push everybody to continue PhD, but she directly, as the fresh graduate at the time, even before doing government tasks, working in a rural area of in Lombok, I think, in Nusa Tenggara. Then she started by requested to be as one of the candidate by LPDP, and then she requests, and then the discussion, and then the LPDP support her, and then after finishing as a doctor Puskesmas, we call doctors in health center in Lombok, she flew directly to Nijmegen. And what happened, and everybody can watch and see that she did very, very, very well. She did a very bright, and she can finish her PhD with many kinds of chapters. I mean, about 9 chapters, and she finished. And then today we have a great achievement of Nadira. Congratulations, Nadira.
Thank you.
I'm proud of you.
Terima kasih, Professor.
Yeah.
Thank you, Professor Ghazem. On behalf of the doctorate board of Radboud University, I'll also congratulate you with your PhD title. And these congratulations are also for your parents, of course, your friends and colleagues, especially the colleagues who helped you with your research, and of course your supervisors. You did a great job already in Indonesia, where you have been very well educated, and now we in Nijmegen could enjoy your talents. Thank you so much for coming over here. I wish you a lot of success in your further career in science, but also in the clinic, perhaps a combination. Well done. We conclude this ceremony with a few words. Please stand up. Gratias tibatimus, omnipotens Deus, pro omnibus beneficiis tuis, qui vivis et regnas per omnia saecula saeculorum.